Hello, everyone. Welcome to another Cambridge Festival virtual event. My name is Anthi, and I'm a researcher at the Department of Chemical Engineering and Biotechnology of the University of Cambridge. Normally, we would be opening the doors of our building today and would have planned a lot of fun activities to guide you in our research world. But it is great that we can still meet in this virtual space to talk science, and thank you for joining me this afternoon. During this session, I'm going to introduce you to one of the most innovative and incredibly useful emerging technologies in the field of biomedical research. We call this technology organs on chips. In this talk, I will try to articulate the working principle of this technology, and through examples of applications, I will attempt to show you how it can benefit studies of human biology, with a particular focus on how this modern technology is shaping up the way we approach drug development. Please use the chat box to ask any questions. I will keep an eye whilst talking, but at the end, I will try to answer as many as possible. Okay, let's begin then. The way we approach drug discovery and development nowadays is an extremely arduous and costly process. As you can see here in this graph, the process for a bright idea to become a drug or a treatment approved and available to patients involves many steps. Every year, thousands of potential drugs are screened, and during the preclinical phase are whittled down to the most promising 10, maybe 20 drugs or leads, as we call them. After thorough testing in preclinical models, maybe 10 of these are taken forward into first in-human clinical trials, during which they are administered to just a handful of participants. Then 2 to 5 of these drugs that show promise in patients are taken forward to larger trials with about 100 participants. And in the good scenario, 1 or maybe 2 or maybe 3 drugs will work well enough to go into late clinical trials with thousands of participants, which will show if one drug works better than a current treatment in order to be approved and licensed. Although each step might be very well designed and the experiments carefully performed, it is clear that the failure rates in clinical trials that test new drugs for their safety and efficacy in humans remain very high. One of the major reasons for such high failure rates is the fact that tools that we currently use to screen candidate molecules before they reach clinical trials have very poor predictive power. In preclinical research, the aim is to determine if a new drug or treatment is effective and safe to use before it can be safely administered to people during human clinical trials. And this stage is very essential for the safety of the human volunteers in medical research. Basically, at this stage, we aim to learn how the disease works, to identify the target of the drug, to determine the amount of drug that can be safely administered to a volunteer while also being effective. Until now, when we are developing drugs, we have 2 tools in our disposal: animal testing and cells in dishes. Well, cells in dishes don't work like they do in the human body, so they're not predictive. And obviously, animals are not humans. A mouse is not like a human being. Although some of the basic bodily processes might be similar, many processes differ immensely between humans and rodents. Let's take, for example, the heart as an organ of interest. The heart of a mouse beats about 300 to 800 times per minute, while the human heart normally beats 70 to 100 times per minute. Because a mouse and a human are so different, a potential medicine might be found to be safe and effective in mice, but is later discovered to be toxic or ineffective in humans. There are also increasing ethical concerns relating to the use of animal studies. Here you just see the principle of 3 R's. which is urging for replacement, refinement, and reduction of animals in research. So the drug development model is broken, and it has created a chasm between a good idea in the lab and the first test of a new drug in humans. That is usually referred as the valley of death here. This valley was also created by lack of funding and drug development expertise on the academic side, but also by concerns about financial risk on the industry side. Luckily, this trip between a bright idea and having a drug in the bench isn't entirely unavoidable. Many ideas cross this gap each year, but the difficulty adds to the time and cost of developing new medicines. As a result, there has been a worldwide search to find replacements for preclinical models that will allow us to better predict how humans will respond to medication. Organs on chips has risen as one of the most promising technologies for enhancing our ability to translate science into innovative medicines. All the efforts in the field are focused on how we can get better translational preclinical data to predict how compounds are going to behave in the clinic. So more compounds will move through clinical development, will get approved as medicines to treat patients. So imagine this technology as a bridge over the valley of death we just described. But great, this potential sounds amazing. What is actually an organ on a chip? Organs on chips are miniature devices that help us researchers to recreate aspects of the structure and function of specific organs in the lab. The word chip in there might confuse someone to think that this has something to do with computer chips. No, these are not computer chips. However, organs on chips have a similar size and are made using similar methods. Nowadays, we can find them in many shapes and sizes, but they share some common features. They contain human cells and tissues, which grow in geometrically confined and patterned compartments within the chips, which we call chambers. They're also connected with pumps, specialized pumps that allow for fluids to flow through the channels and reach the chambers where cells grow and provide them with the necessary nutrients and gases like oxygen. And even this flow is used to mimic the blood flow in our body. Chips also contain sensors that help us to tune and control the environment of the chamber according to the needs of specific cells, but it also allows us to obtain readouts to control the exposure to the therapeutic compound, but generally also to see how cells grow and if they are healthy and functional. The fluidic flow also allows us to obtain samples for further analysis in the lab. Now let's see in more detail how this works. So in my perspective, organs on chips is a new way to mimic human biology. The traditional way of doing so is looking at a single cell or clusters of cells in a dish. But organs on chips are trying to shift this perspective and look up from a bigger system view. So how we can recreate biological systems in a way that has meaning back to the human whole body. Each chip is engineered to create the right environment for cells to feel like they were in the human body. So the first component for an organ on a chip, of course, is cells. Um, our body is full of organs. You probably know that each organ is specialized for a different function. If we are to use the heart example we mentioned before, our heart pumps blood through the body. Almost all our organs consist of many smaller parts that are called functional units. And functional units are made of different types of cells that are very, very well trained to perform their assigned tasks. Again, with the example of the heart, its functional unit is a fiber, which is made from a few heart muscle cells that rhythmically contract. These fibers bundle together to help the heart to pump blood throughout the entire body. Thanks to new discoveries in cell biology and other novel technologies, it is now possible to grow those functional units as mini organs outside the body. We have various cell sources at our disposal, but stem cells are without a doubt the most important source. Stem cells are very special cells. They can both replicate themselves as often as they want, and at the same time, we could shift them, we could help them to change into any other cell type in our body. And this process is called differentiation. A few years ago, scientists found a way to turn skin cells, meaning already specialized cells, back into the state of this stemness. And then other scientists and other groups managed to convince these stem cells to respecialize even into any other cell type, for example, heart cells or liver cells. And this was a breakthrough discovery that led to another major discovery for biomedical sciences: organoids. Organoids are three-dimensional cell structures that contain a multitude of organ-specific cells. We get those miniature organ-like structures through the differentiation and self-organization of stem cells, isolated from various sources, even from our own body, and then we treat them with cocktails of very specific molecules and growth factors that direct them to specialize towards the cell type we want to study. Now, organoids and organ chips are not the same thing, um, but we use organoids as cell sources that better represent cellular environments found in our bodies, and they are better are doing so compared to the conventional cell cultures in a dish. The ability to self-organize helps organoids to mimic the general tissue structure and our developmental trajectory, and this can often give us cell types that would have otherwise been very difficult to maintain with traditional methods. Now, how does this work? How putting cells in an organ on a chip works? These miniature devices are usually made from polymers. Polymers are huge molecules that contain long chains made from many of the same small molecule. Plastic is an example of a polymer. For us, for organs and chips, the most common material is PDMS, which is transparent and flexible, kind of like a plastic. But one could say that the devices, these organs and chips, look like and are the size of Lego bricks. As we mentioned before, each device may contain many small channels and chambers where cells grow, and the fluids that nurture them flow via dedicated ports with the help of specialized pumps. Because these channels and chambers are so small, and because we can flow through them liquids or gases, the polymer bricks are called microfluidic platforms. So this is another term you might hear when we're referring to organs on chips.
Wow, this
Here
is
in
fucking
this
crazy
graph, we
shit.
have two chambers in this
This is
organoid chip.
like
They're
genuine
separated by
space age
porous
malarkey, isn't it?
membrane,
Come on.
or we can say in other words that the chip comprises multiple layers, and here the top and the bottom layer sandwich a membrane. The microenvironment formed in its chip provides a cozy environment for cells, helping them to forget that they are not in the body anymore. And it allows cells to arrange themselves in their accustomed configurations, interact with the neighboring cells, similar to the way that they would in the human body, forming functional units. That's why I call this home away from home. Once settled down, the cells continuously feed on sugars and other nutrients that are dissolved in the liquids, pumped in through the channels by the dedicated inlet ports we saw before. These channels are next to the cell chamber. So this is the cell chamber, this is the channel. And in this way, in organs on chips, we can mimic the way blood is flowing through blood vessels surrounding the organs. The nutrients can squeeze through tiny pores between the channels because of this membrane we mentioned before, but the cells cannot escape the chamber. Similarly, the flow can transport waste products produced from cells and other messenger substances away from this chamber. Um, in this figure, the chambers in the bottom layer contain cells that are continuously supplied with fresh nutrients from the channel above. Finally, because in most cases the material that chips are made of is transparent, the cells can be viewed with the microscope through a tiny window on the bottom of the chip. So this is how this works, and the benefits of this technology and how its adoption has a genuine impact in the drug discovery field, but also in a wide range of research fields. These are all highly valued now by research institutes and pharmaceutical industry. This might not be the most updated graph, but you can get the idea. The organ on chip community is growing fast, and we now have numerous chips tailored to mimic specific organs and, in fact, specific functional units of each organ. There are numerous applications and ways we can use these organs on chips. I have cherry-picked some of them to use as examples of how this technology works and what it can do for us. But bear in mind, these are just a few examples. So let's start with the example of designing a lung on a chip model. The first step is to understand the anatomy of the target organ and reduce it to the basic elements essential for physiological function, what we called before functional units. Then each functional unit is examined to identify the different cell types, how they organize, and what organ-specific signals are necessary for the function. Here, the alveolar sac of the lung consists of alveolar epithelial cells, cell type 1, and microvascular endothelial cells, cell type 2, that are closely opposed to each other, and they're separated by a thin layer of connective tissue. Now, the epithelial and endothelial layers are subjected to air and blood flow, respectively, and the multi-layered interface experiences mechanical forces induced by breathing motions. This is the organ-specific microenvironment description. Now that we have identified the functional unit features and the structural organization, we have to design the device accordingly. This lung on a chip system consists of two overlapping microchannels that sandwich a membrane that is thin, flexible, and has tiny pores, so it can enable communication between these two cell types. This design nicely mimics the structure of the alveolar sac, and at the same time, we can have a co-culture of these two types on either side of the membrane. In addition to that, the cells are exposed to their respective tissue-specific microenvironment. So air is pumped in the chamber of the epithelial cells, and fluid mimicking the blood flow is perfused through the endothelial side. So the fluid should be in here and the air should be pumped in here. But we also said that the alveolar sac experiences mechanical forces as a result of breathing. To mimic this, the device is equipped with two hollow microchambers alongside the culture channels that we can mechanically stretch, and this stretching of the membrane that contains both cell types is mimicking breathing motions. Another example is the human blood-brain barrier on a chip, BBB on a chip. This is a very unique and a highly specialized tissue that acts as a selective barrier that controls transport of molecules from our blood to our brain. And it's trying to maintain physiological conditions for optimal function of our central nervous system. The BBB is composed of several cell types that collaborate to form this tight barrier that, along with the surrounding extracellular matrix, imagine this as a gel-like tissue that lies between our organs. So along with ECM, extracellular matrix, these special BBB cells create a microenvironment crucial for optimal function. Now, neither animal models of the blood-brain barrier nor conventional cultures of cells in a dish alone effectively mimic the barrier and transport functions of the BBB observed in our body, in our human systems. Thus, there is a great need for a human blood-brain barrier model that could be used to develop new and more effective therapeutics so they can be safely administered, safely delivered to the brain, as well as having these models as tools to deepen our understanding of disease mechanism. Now here in this example, the researcher used a device that comprises, again, two chambers separated by a thin flexible membrane. One side of membrane is lined with brain endothelial cells, and the other chamber the endothelial cells are here, and then the other chamber is lined with specialized cells we call astrocytes and pericytes, and of course the relevant fluids are pumped through each chamber. What was really cool in this example is that the researchers also managed to control the oxygen levels inside these chambers to mimic the oxygen levels we have in the human body. And this fact was shown to significantly improve the morphology and the function of this blood-brain barrier model. Then they used these chips to validate transport mechanisms of drugs and other therapeutic molecules across this BBB. In particular, they investigated a series of transport mechanisms that either prevent drugs from reaching their target to the brain, so they pump them back into the bloodstream, or in contrast, they allow selective transport of nutrients and drugs across the blood-brain barrier. Understanding those will facilitate development of new approaches to deliver drugs into the brain, and thus help a lot of patients suffering from neurodegenerative diseases and neurological disorders. Another example of organs on chips application is modeling cancer. And here I have the example of pancreatic cancer, which is one of the most insidious forms of the disease. I think on average, only around 10% of patients are alive a few years after diagnosis. And one of the reasons for such a dismal outcome is that pancreatic cancer cells are able to escape from tumors and enter the bloodstream very early in the disease. This means that by the time the cancer is discovered, it has already spread. Paradoxically, pancreatic tumors appear to almost lack blood vessels altogether, which prevents cancer drugs from reaching and killing those tumors. This has been puzzling scientists and clinicians a lot, who are still trying to understand how the disease progresses. Well, let's see how this device now can help researchers towards this goal. In this device, we have again a clear, flexible plastic chip about the size of a USB stick. The chip in this case is filled with collagen, and in here we have two microfluidic channels patterned inside this collagen, and each of which can be lined with living cells that are kept alive by a constant flow of nutrient-rich media. One channel mimics the blood vessel, and the other channel is a pancreatic tumor. Now, what researchers found with this device is that tumor cells invade nearby vessels, destroy the endothelial cells that line them, and replace those with tumor line structures. So you can see here tumor cells invading the blood vessel. In addition, the group identified with this chip, with this study, the interaction of a specific protein in pancreatic cells, in cancerous pancreatic cells, with a receptor on the surface of the endothelial cells as the driving mechanism for cancer metastasis. This was very surprising. And they suggest that this could be used as a target to develop future treatments. For example, if we have a drug that inhibits this interaction, then we could rescue the blood vessels from cancer invasion, and probably we could deliver drugs to shrink down the tumor mass, which currently is impossible to do. This organ-on-chip application really exemplifies the power of this technology to replicate disease state in the lab, to identify drug targets and mechanisms of action in a way that is superior to preclinical research tools. Let me now show you an application developed by our group. In the Bioelectronic Systems Technology Group, we use organic polymers, which are electroactive. So we use these materials as the building blocks to design various devices. Some of these can work as organs on chips. And we use these materials to interface them with biological systems that we also develop in the lab. Now, I said that this material is electroactive. This means we can use this material as an integrated cell sensor. Basically, when cells grow in the material, they change the properties of the material. Okay. And by monitoring these properties, these electrical properties of the material, we can obtain information relevant to the activity of cells, if they're healthy, if they're dying, if they're functional, if they're becoming the tissue we want them to become. This is very useful for building organs on chips because it enables us to get real-time and continuous information about how cells behave in the chip. An example of that is our bioelectronic gut on a chip. We can see here the structure of the gut. Essentially, this organ is a tube that has very thick walls that consist of multiple layers. And its role is to be the gatekeeper of our body. It's making sure that all the nutrients, vitamins, and other essential molecules for our health will enter our body. But at the same time, it keeps out pathogens and toxins, as well as the microorganisms that reside on the surface, in the gut lumen, which is known as the microbiome. To do that, the gut is equipped with a layer of several types of epithelial cells that form a selective barrier between the gut lumen and the bloodstream. You can see here a tiny blood vessel. So having identified this function of the gut as the functional unit of our organ on a chip and taking into account the tubular architecture, we developed a 3D organ on a chip and we named this Hubister. Here you can see in dark blue, black, our polymer, our organic polymer, which I said is electroactive. And we use this and put it inside a tube. That's where Hubister comes from. And we also patterned a hollow channel in the middle to mimic the gut lumen. I'm not sure if you can see here, there are also two gold electrodes around the polymer scaffold, and this helps us establish contact with the material so we can measure the electrical properties at any given time point. Then we seed intestinal epithelial cells to mimic the gut barrier, but we also use, so here is the gut barrier, but we also use fibroblasts to mimic the interface between the gut barrier and the blood vessels. After almost a month, we see the cells forming a structure very close to the one in our body. Because the soft polymer we use is electroactive, it can conduct ions and electrons, We use this as an electrode inside the system to sense how cells proliferate and form the intestinal tissue. And by evaluating these electrical readouts, we can almost obtain them daily, we can characterize the integrity of the intestinal barrier. Now, the integrity of the intestinal barrier is a very important parameter for a healthy and tight and functional intestinal barrier. This barrier is target for some pathogens and toxins. Upon inflammation, for example, this barrier is significantly damaged. We have now data showing that we can use our bioelectronic gut to model situations like inflammation, and potentially we could test drugs or treatments to heal inflammation, but also we can test if other drug candidates and see if they can cross the intestinal epithelium, reach the bloodstream, and eventually arrive to the target organ in our body.
Okay.
While organs on chips
Okay.
are of great use for both organ level and fundamental cell
What's
biology
this child
studies,
saying?
such as pharmaceutical testing, the greatest potential of this technology is revealed by connecting multiple organs on chips in one platform. So we're going now towards multi-organs on chips. Such platforms would allow tissues to grow independently while accurately mimic the function of human organ, but at the same time, the organs can interact with each other, simulating the way organs communicate in the
This is
body.
how it
Now,
begins.
currently, there are two approaches for building multi-organs on chips. First is by
Androids gonna
linking
take over the world.
organ
You
chips
watch.
together
This
via
is how they're gonna
their
build them.
fluidic channels.
Microchips.
And the second is growing different organs in the same chip, which are again connected via specific microfluidic channels, but in one platform.
See,
Now, why is this useful
human
and
on a
how can
chip.
it benefit drug testing?
Android
By joining
aliens
together
taking over
different
the
organs in
world.
one single network, we can follow a drug through its journey to reach its target through a simulated human body. Let's take the example of, let's say, an aerosol drug, like the one dispensed by an asthma inhaler. In a multi-organ on a chip platform, we could monitor in real time how the drug enters the lungs, how it affects the heart, how it is metabolized in the liver, how it is excreted by the kidneys, and if there are any side effects along this journey. Let's see some real applications. Uh, One example where living animals must be used in preclinical testing is the characterization of a drug's pharmacokinetics. That's a strange term, but it refers to the quantification of the drug absorption, distribution, metabolism, and excretion. And all these data together help us determine the level of drug in the blood. Animals are also used to analyze drug pharmacodynamics, another tricky term, but through that, we can determine the effects the drug produces on its target organ, which means we can see what is the mechanism of action of the drugs, and we can also identify and measure adverse effects. All these responses involve interplay between many different organs in our body and the bloodstream. Or, in other words, we need the whole organism response to determine these pharmacokinetics and pharmacodynamics. Weird terms, but that's how we call them. And until now, animal models was the only way we could run such studies. Now, in this example here, this is a multi-organ system. The research team was able to create a functional platform to obtain experimental data that, together with computational modeling, was able to generate data about both pharmacokinetic and pharmacodynamic behavior of drugs in a human system. So here on top, the first application, what they did was to add nicotine. So the drug here is nicotine. This, besides helping with smoking cessation, it is also investigated as an oral drug, I think, for neurodegenerative and inflammatory bowel diseases. So they added nicotine to the gut chip. in the epithelial channel in this first top to mimic oral administration of the drug, but also to mimic the transport from the intestinal wall to the bloodstream here in this channel, and from there how it goes to the liver where it is metabolized and how it goes to the kidney where it is excreted. And this was done here also by the researchers also took samples in the same way we go to the clinic and get a blood test, and this helped them, along with the computational modeling, to translate the data from the organ chip scale to the actual organ dimensions in the human body. And they saw that the platform and their experiments were matching findings of human clinical trials. So this is a very good sign that organs on chips can be implemented in clinical trials, in drug testing. They also tested cisplatin, which is a chemotherapeutic drug commonly used in cancer treatments. This one is administered, I think, intravenously, but it displays a lot of unwanted toxicity in the kidney and in the bone marrow. So they changed a bit their platform here, and they have a bone marrow model, a liver chip, and a kidney chip. The experiments also produced information about how cisplatin is metabolized and cleared by these organs, and they produced results that are similar to human body responses. So we can see in this example that we can get accurate preclinical testing of drugs, obtain valuable information about the human body outside the human body and without the use of animal models. Recently, another multi-organ platform was developed that was also able to link together up to 10 different organs. It's called Physium on a chip. Although the tissues don't replicate the entire organ in here, they do perform many of its important functions. Here you can see the general design, but each organ model can be scaled up or scaled down, and the operation principles can be tuned to accommodate the requirements of its application. So according to the question we're asking this model, we have to make sure that we change the parameters in order to be able to have the answer we're looking for. So using this system again, the researchers showed that they could deliver a drug to the gut, mimicking oral administration, and then observe the drug as it was transported to other tissues, it was metabolized, etc. And they would also measure where the drugs went, which organ chips in this platform it reached, the effects of the drugs on these different tissues, and how the drug was broken down. This kind of system holds great potential for accurate prediction of complex adverse drug reactions on human systems. So I think it's a new approach in drug discovery, and we can see this happening. In our group, we're also working on multi-organ systems. In particular, we're trying to build a platform using the gut, the bioelectronic gut I showed you before, and mimic the way the gut communicates with the brain and the brain communicates with the gut. We call this gut-brain axis. And we want to use this platform and provide researchers with a tool to test drug candidates for intestinal and neurodegenerative disease. But what we're really interested in is to study how this complex community of microbes that resides in our gut, we call them microbiota or microbiome, and we want to see how these affect our health, if and how they're involved in diseases that affects other organs and particularly the brain. And we hope we're going to use this 3D organ on a chip as our Lego brick to build our platform. Now, in summary, I hope that I convinced you that organ on a chip technology is a new, better way to mimic human biology. Until now, we could look at cells in a dish and examine patients and individuals in clinic. But there was a missing link in between, and organs on chips have risen as a highly promising tool that opened this window for us to look into human biology. These chips allows us to see biological activities in human cells
I
and
still
tissues of
think
organs that
it's a
we
little bit
probably did not
suspicious,
know
isn't it?
before,
We just want to put these
including
little
things that
microchips
happen in
on
humans
the
and not
inside of you.
in animals. This is particularly important for drug development
Don't need
studies
androids. Just gonna
and
put a
at
chip
this
on you and
stage of
take over.
valley of death we described earlier.
And the
This means we could
government
test
will
drug
will
candidates
tell
in
you what
these
to do.
chips to
It's
study
like,
whether the medicine works and or if it does any
I
harm
mean, it's a possibility.
specifically to the human body, and decide if it's
Like with a
safe,
lot of
if it's
things, if
safe to
those in
move.
power get the option
Next
to do
stage
shit like that, then
drug
they
development
will.
process and test this
So
in
you have to
human
watch for them
volunteers
buggers.
with efficacy and safety.
Unless you're one of the people
By
in
doing
power, in
so,
which
we
case you just
also
got to
would not
watch
need
for
to
the
use
reason.
laboratory animals anymore to know if medicine is safe. So, animal testing can be progressively reduced over time using organs
I
on
do like
chips.
this because
There are quite a
anything that
few
reduces
pharmaceutical
animal testing is
companies
a
that are using
great
organs
idea.
on chips coming out of research institutes around the world, including Cambridge University. And it has been shown that we get completely different results between animals and chips. Most people in science are so inbred with using animal models, and in some cases, we forget that we don't actually predict what happens in humans when we use animal models. And we are in a loop where we don't believe that there is an alternative.
Yeah,
And
I mean,
this
this is
technology
the thing.
offers an alternative. The more the people become familiar with the possibilities of organs on chips, the better a chance we have to get out of this loop, to deliver drugs faster,
So
greater
using
accuracy,
animals for
and at lower
mechanical injury
costs.
training and stuff
Now,
is
the next
probably
level
pretty
of organ
sensible,
on
but
chip involves more than finding
for
the
the
right medicines
biology,
for
as you
humans.
say,
It
and the
could be used to
physiology,
find
it's
the best
different.
medicine for each individual
So
person.
everything
So,
works different.
your body on a chip. People differ in the ways their bodies handle diseases and react to medicines. Just because a certain medicine at a certain dose might help you treat a disease does not automatically mean it will help your friend too. In the future, we might be able to painlessly donate cells to build personalized organs on chips, your body on a chip, a model that will represent just us as individuals and test whether a specific medicine will work for us and us only. Now, with that, I will stop bombardizing you with information. Thank you
Bombardizing.
all for listening. While I check if you already have posted questions in the chat, but also give you time to type your questions, I will put on this slide. These are all events our department is running during the next few days as part of the Cambridge Festival. My colleagues have put together some really interesting and insightful talks and fun activities that are not to be missed. So let me check if we have any questions in the chat. I see already you have some. So if I think that the organ on a chip can be too simplistic to recapitulate the complexity of human body, yes, the models are um, simplistic and are reductionist intentionally. We want to focus specifically every time on these functional units we identified. But in the future, we might have this ability to do that better. So as the technology progresses, um, both in the from the standpoint of biology, so we have more robust cells, but also from the standpoint of technology, we will improve that. We will never have um, the full complexity of a human body. So, for example, we will never be able, I think personally, we will never be able to model consciousness. We won't be able to take into account what gravity does in our body. So it will recapitulate a lot of the complexity, but not the entirety. Even the fact that we can do this at the moment at this level means is already a great step. And I think in the future we will see this technology being more integrated. Hope this was helpful. Let's see if there are other questions. How do we validate that the cells behave in a similar way to inside the body? There are various other assays that we run. I mentioned before that most of the organs on chips have a port window in the bottom. So we use microscopy and we can see the cells through a microscope and identify their morphology, the structure that they build together, if they express specific biomarkers that will help us understand that this is functional and healthy. That is a cell that very well represents a similar cell in the body. There are also other assays, molecular biology assays, because we have these fluidic channels and the ports, we can get a sample of the fluid that has passed through the chamber of cells. In the same way, we would give blood to the GP and get tested to see the levels of iron, for example, in our blood. We could run similar tests and see if the cells have secreted specific biomarkers. If we're testing, for example, for inflammation, we could see if there are any markers related to that expressed as the fluid goes out of the cell. Any other questions? I think I answered this one, so I don't think brain on a chip will have consciousness. I don't think this is going to happen. Have an indirect beneficial impact in tissue engineering research. So can the research using organs on chip have an indirect beneficial impact in tissue engineering research? And if we use biopolymers as scaffolds or a hybrid between organic and synthetic materials? So definitely organs on chips are used for tissue engineering purposes, both in vitro engineering and as preclinical testing for in vivo engineering. We use personally in our lab, we develop models within this technology that are trying to mimic as best as possible tissues and study, as I mentioned, disease, even a healthy tissue, how it behaves, how cells communicate with each other. Do one cell type helps another cell type perform a function better? We can test the effects of drugs. We could even test the effects of nutrients, so something that is not harmful in the body. We can see what is happening. And I know that people who are using organs on chips to test, for example, various approaches and treatments for regenerative medicine. So it's a tool that it benefits tissue engineering before this is reaching the clinic. And because these organs on chips are really, really better at mimicking human biology, we can get results that can be moved to the clinic faster. And if we're using biopolymers as scaffolds, yes, we do. And we also do use hybrids between them. So the materials we use, the organic polymers, are synthetic materials. They're not natural materials, but there are numerous applications in our lab and in other labs where you can blend collagen, for example, or hyaluronic acid, which are natural biopolymers. These are found in our body. You can blend them with synthetic materials. And you can do that always keeping in mind what is your goal. Are there any other questions? Maybe I'll give you a few minutes